Welcome to Dr. B's tutorials. Quick heads up, if you stay till the end of this video, I'll show you the cancer biology questions that show up most often on AP and IB exams, plus the mistakes that quietly destroy scores every year. Your future test-taking self would like you to keep watching, so keep watching till the end. Cancer is not just one disease, which already makes it suspicious. Biologically, cancer is uncontrolled cell division caused by failures in regulation. These failures come from mutations that accumulate over time. The cell cycle normally has rules, checkpoints, and supervision. Cancer cells ignore those rules completely. Instead of stopping when told, they keep dividing. Think of cancer as regulation breaking down, not cells trying to be evil. Normally, cells do not divide randomly. They move through the cell cycle in a specific order. G1 is growth, S is DNA replication, G2 is preparation, and M is division. Progression requires internal and external signals. Cells check for DNA damage and proper size. If something is wrong, division stops. Cancer begins when these controls fail. Checkpoints are quality control stops in the cell cycle. The G1 checkpoint is the most important for cancer prevention. Cells check size, nutrients, and DNA damage here. The G2 checkpoint ensures DNA was copied correctly. The M checkpoint checks chromosome attachment. If problems are detected, division halts. Cancer cells blow past checkpoints like stop signs at 3 a.m. Cyclins and CDKs are the molecular managers of the cell cycle. CDKs are enzymes that push the cycle forward. Cyclins activate CDKs by binding to them. Cyclin levels rise and fall throughout the cycle. This ensures timing is controlled. When regulation fails, CDKs stay active too long. That is bad news for cell division control. Proto-oncogenes are normal genes that promote cell division. They are essential for growth and development. Under normal conditions, they respond to signals appropriately. When mutated, they become oncogenes. Oncogenes are stuck in the on position. This causes excessive division. Cancer loves gain-of-function mutations like these. Tumor suppressor genes are the brakes of the cell cycle. They slow or stop division when problems occur. Many help repair DNA damage. Others trigger cell cycle arrest. When these genes fail, division continues unchecked. Cancer often involves loss of function mutations here. Losing the brakes while flooring the gas is a terrible combination. P53 is the celebrity of tumor suppressor genes. It is often called the guardian of the genome. P53 detects DNA damage. It can stop the cell cycle to allow repairs. If damage is too severe, P53 triggers apoptosis. This prevents damaged cells from dividing. Over half of human cancers involve P53 mutations. Apoptosis is programmed cell death. It is an organized and intentional process. Cells that are damaged beyond repair are eliminated. This protects the organism. Cancer cells often avoid apoptosis. Avoiding death gives them a survival advantage. Unfortunately, this allows mutations to accumulate. Cancer does not usually result from one mutation. It requires multiple mutations over time. These mutations affect different regulatory systems. As control erodes, division increases. More divisions lead to more mutations. This creates a dangerous feedback loop. Cancer is a process, not a moment. Cancer cells divide rapidly and mutate frequently. This creates genetic diversity within tumors. Treatments may kill some cells, but not others. Resistant cells survive and multiply. Many cancer treatments also harm normal dividing cells. Regulation pathways are complex and interconnected. This makes cancer biology incredibly challenging. AP and IB exams love cancer questions. They often ask you to compare proto-oncogenes and oncogenes. Tumor suppressor gene function is a favorite. P53 appears constantly in scenarios. You may need to predict outcomes of mutations. Data interpretation is common. Explaining mechanisms earns points. For exams, always explain how regulation fails. Use precise biological terms like loss of function and gain of function. Link mutations to cell cycle effects. Avoid vague answers like cells divide more. Think in cause and effect chains. Drawing pathways can help organize ideas. Cancer questions reward deep understanding, not memorization. That's it for now. Don't forget to subscribe, like, and check out Dr. B's tutorials on Amazon for full AP and IB study guides. See the first link in the comment below. For more tutorials and test prep videos, subscribe to our YouTube channel, Dr. B's Tutorials.